Hello, my name is Negrin, and I've been working at the Lázaro lab all summer on this exciting project. My project is entitled Investigating the Translation Efficiency of Modified mRNAs in Different Cell Types. So a little bit of background going into this, I really want to highlight cardiomyocytes. They are heart muscle cells responsible for various vital heart functions such as contraction and relaxation. Unlike other cells in our body that keep regenerating and keep dividing throughout our whole lifetime, cardiomyocytes shortly lose their ability to divide after birth. So if we suffer things like heart failure, other cardiac traumas like heart attacks, that cell is permanently damaged around our heart muscle. So how do we fix this? So we use a technique called cell reprogramming. We use the overexpression of 4 transcription factors. I specialize in OCT4 during this experiment. We also utilize mRNA as an ideal way to express these transcription factors to bring it as an intermediate step, as you can see in this picture on the right, and bringing it back to really a partially reprogrammed state, not all the way, as it is a transient molecule. And then we just want to, again, just return cardiomyocytes to their healthy state. So the purpose and significance of this project is that messenger RNA holds a great promise for gene therapy due to its fast but transient expression. However, mRNA is inherently immunogenic and unstable. Nucleotide modifications can help with the stability of this, and that's what I have been doing, adding nucleotide modifications to all of my RNAs. And this has been implemented already into the FDA-approved vaccines against COVID developed by Moderna and Pfizer. So it is a promising field. Not going to go into in-depth the DNA protocols, but we do use tail PCR, and then we purify the DNA until we get into this purified and concentrated state. And then we move on with RNA synthesis, which we use in vitro transcription to add the nucleotide modifications. As you can see right here, these are the modifications. And then we also add a capping agent. And then we purify it using ethanol precipitation. And then these are all of the modifications that I did in my project. So we did unmodified, 5-methoxyuridine, and 1-methylpseudouridine, 5-methylcytosine, and a combination. So, next with the methods, we also measured concentration throughout this whole project using nanodrop and qubit. We also ran gels using gel electrophoresis to confirm the size of the plasmid RNA. We used DNA ladder or RNA ladder to really cross check the size that we were expecting, and we also cultured AC10 cells, which are cardiomyocytes, and used them for transfection. So this is a really nice picture of my flask with AC10 cell line. So our results. First, with gel electrophoresis, this depicts all the concentrations of all the modifications. At the time, we had adjusted them, so they should have all been the same concentration, and we believe that they were not showing this so because we have been running into issues with accuracy and reproducibility using the nanodrop or qubit for telling your concentration comparisons. So this is our chart depicting the comparison of the concentration measurement accuracy of both methods. But then another experiment that we were also running was that we transfected the cells. Basically, transfected the cells with different ratios of mRNA to lipofectomy. Lipofectomy is a transfection agent. And this was the best dose that looked best for the cells and was most consistent. This ratio of 0.2 micrograms of mRNA to a high dose of lipofectomy. And so our conclusions are that AC10 is most effectively transfected with lipofectamine. We are still undergoing investigation on what is the best method to figure out mRNA purity and best quantification method for concentration. And to repeat the steps, we would use the different transcription factors. I only started with one this summer, and investigate if these findings are comparable to other cell types, if this research is cell type specific. And this is an acknowledgement to my lab